Good evening, everyone. Myself, Simran. Today, I am going to present applications of transformation in microbial genetics. Contents: Introduction, what is transformation, application of transformation, and conclusion. Introduction. What is transformation? Transformation is the process in which the genetic material is transferred from one bacteria to another bacteria without involving other organism. A bacterial cell acquire DNA from their environment. This process is called transformation. Transformation was first discovered in bacteria by Frederick Griffith in 1928. He suggests that bacterial strains are capable of transforming themselves by some factor which he termed transforming principle. There are two types of transformation: natural transformation and artificial transformation. In natural transformation, bacteria naturally have the ability to incorporate DNA from the environment directly. In artificial transformation, the competence of the host cell has to be developed artificially through the different techniques. Application of transformation in microbial genetics, in DNA cloning, genetic engineering, gene therapy, and chromosomal mapping. What is DNA cloning? DNA cloning is a molecular biological technique which is used for the creation of exact copies of a particular gene or DNA. DNA cloning is the process of making of multiple copies of a particular segment of DNA. During this technique, the selected DNA fragment is inserted into plasmid using enzymes. Restriction enzymes and DNA ligase are used in this process. DNA cloning can be used to make proteins such as the insulin. It is used to develop recombinant version of the non-functional gene to understand the functioning of the normal gene. It helps to analyze the effect of mutation on particular gene. Genetic engineering. Genetic engineering is the process of direct manipulation of organisms' genome using biotechnology. It is a set of technologies used to change the genetic makeup of the cells, which includes transfer of gene within and across the species boundaries to produce an improved or novel organism. It is used in the manufacturing of drugs, in human growth hormones, and the production of monoclonal antibodies and the preparation of vaccines. It can be used on various plants, animals, and microorganisms. Genetically modified crops are produced using genetic engineering by using these techniques in agriculture. The crops provides a protection from insects and pests. Gene therapy. Gene therapy is a technique which involves the replacement of defective genes with healthy ones in order to treat genetic disorders. It is used in the replacement of genes that cause a medical ill health. This method is generally destroys the problem causing genes. It helps the body to fight against disease by adding genes to the human body. This method is useful to treat diseases such as single gene disorders like cystic fibrosis, sickle cell disease, and complex diseases like cancer and HIV. Chromosomal mapping. Chromosomal mapping involves the identification of the location, structure, and function of every gene that is present on each of the chromosome present in the nucleus. By using this chromosomal mapping, the Human Genome Project is a wide successful genomic attempt by scientists to map all the genes that are present in the human chromosome. This technique is used to identify the chromosomal mutation. In conclusion, we have explored the fascinating world of genetic microbiology. We have seen how transformation allows to edit genes, develop a new therapies, and create a genetically modified organism with incredible precision and efficiency. As we look to future, this techniques opening up a new avenues for the research and innovation. References. Thank you.
Thank you, Simran.
Thank you, Sid.
If there are
Thank you,
no
Sid.
presenters today, I wish you all a very good evening. Have a nice time. See you tomorrow.